Good morning or afternoon, everyone. I am Bruna Lousada, the European Manager at Hansen, the Teledyne Labs division specialized in dissolution and diffusion test systems. And I want to welcome all of you to today's webinar titled Understanding the EMA Guideline on Quality and Equivalence of Locally Applied, Locally Acting Cutaneous Products and the Implications for IVRT. With the EMA updated guidance that went into effect in April of this year, this is a timely and important topic for everyone involved in the development, testing, or regulatory submission of topical formulations, as the expectations around demonstrating quality and equivalence are evolving rapidly. Today's session will help clarify what's changing, what's expected, and how to navigate these requirements effectively. So we are thrilled to be joined by Sasha Gorison. He's the co-founder and head of laboratory at Rades, a Hamburg-based company specializing in contract formulation and development. Rades was founded in 2018 by a team of seasoned pharmaceutical experts and has quickly built a reputation for excellence in semi-solid and liquid dosage forms. The rational design approach allows them to tackle even the most complex formulation challenges with precision and efficiency. Sasha brings over 25 years of experience in the pharmaceutical industry with deep expertise in preclinical development, pharmaceutical analytics, and project management. His insights today will be invaluable for anyone looking to understand how to apply the new EMA guideline in a practical and scientifically sound way. So before we dive in, a quick note, all attendees are currently muted, but feel free to drop your questions as we go using the question function on your screen. We will address as many questions as possible at the end of the presentation, and if we run out of time, we will follow up via email. If you experience any issues with your audio or connection, please try logging off and logging back on. For those who can't watch the entire webinar or wish to revisit later, a replay will be available on the Teledyne Labs YouTube channel in the coming days. So with that, let's get started. Sasha, over to you now.
Yeah, thank you, Bruna, for the kind introduction, and welcome everybody to the webinar. In today's webinar, I reflect the implications of the new EMA guideline on quality and equivalence of locally applied and locally acting cutaneous products on the requirements for in vitro release testing. Initially drafted in June 2018, the revised version became finally effective in April this year and applies, as the title mentioned, to locally applied and locally acting drug products, but can also be relevant to auricular and ocular products, as well as for products intended to be applied on the nail or to the mucosa. So, why do we need this guideline? We need this guideline because any changes in the drug product composition, in the dosage form, in the manufacturing process, etc., may have an impact on the efficacy or safety of your product. And it is up to you to prove that this is not the case. So therapeutic equivalence studies with clinical endpoints are in principle necessary to prove this. But as we all know, this kind of studies are typically time-consuming and expensive, and other models are of interest to overcome this hurdle, and this is the intention of the guideline, to give you support and guidance on how to use alternative methods like IVRT and IVPT to substitute clinical data for the demonstration of therapeutical equivalence. The requirements are quite high and based on the assumption that you have a high quality drug product. To ensure that, the guideline is separated in two major parts. The first part is the quality part, which is relevant for all new products, generics, and for post-approval changes. And the second part is equivalence, which is relevant whenever you have to compare two products to each other, maybe for the development of a generic product after a post-approval change or for scale-ups. In order to guarantee a high-quality product, it is, from my point of view, absolutely essential to know the properties of your product. And drug release is considered by the authorities as a critical quality attribute, which needs to be specified and controlled. That sounds easy, but may have a significant impact on the development, because the specification limits should be referenced to clinical batches. But this means you have IVRT data in place from your clinical batch. Do you have this data? Or in addition, the guideline suggests to implement IVRT in the stability program. Have you thought about how this could be implemented? And be aware that IVRT is also necessary for scale-up. So the key messages of the quality part are: you have to well understand your drug product. The drug release is a critical quality attribute which needs to be specified and controlled, and the specification limits based on your pivotal clinical batch. This is very important. And IVRT as a drug performance test is part of the stability program if this is a relevant product characteristic. And IVRT is necessary for commercial scale-up. From quality to equivalence, to therapeutic equivalence. Before we start, we should go quickly through some definitions here. So therapeutic equivalence means that the efficacy and safety profile of a test and a reference product is sufficiently comparable so that no clinically relevant differences can be observed. So the focus here is on no clinically relevant differences. Besides this, a simple formulation in the context of the guideline is a single phase formulation without any excipients which are intended to enhance drug permeation or are difficult to characterize. And when we have a simple formulation, we also have a complex formulation, and the complex formulation here is a multiphasic formulation like an emulsion, or a formulation which contains penetration enhancers or excipients which are difficult to characterize. So this definition is important for the guideline. The demonstration of the therapeutic equivalence by alternative methods is a stepwise approach, and it consists of up to 3 levels. First, and basic level is the assessment of pharmaceutical equivalence. In Q1, you assess the excipients if the excipients are qualitatively the same. In Q2, you ask for the similar amount of the composition, if the amount of the composition is similar. And in Q3, whether the arrangement of matter, like rheology, is comparable. This is a quality part of your drug product. In the next level, the drug skin permeation will be assessed using different tools like PK, IVRT, or tape stripping. And the selection of the tools is depending on the permeability of the API. This is the biological part. The third part is a clinical study it should be used whenever the requirements of step 1 and 2 are not fulfilled. As mentioned before, the demonstration of the therapeutic equivalence is a stepwise approach, and IVRT is important, but only a part of this process. In the easiest case, you have a clear solution without penetration enhancers and any complex excipients. Then you can easily demonstrate the therapeutic equivalence just by fulfilling the requirements of level 1, the pharmaceutical equivalence only. For all other conditions, you have to conduct an IVRT study, except for the conditions that Q1 and Q2 and Q3 are not similar. But to be honest, then it is a different drug product and you have to perform a clinical study. That's clear. Important to note here is that the results of an IVRT can be overridden by the next level test. So, for example, by an IVPT study. This based on the assumption that the observed differences in IVRT are over-discriminative and maybe not clinically relevant. Post-approval changes are very common during the life cycle of a drug product, and the activities based typically on a risk assessment. If you identify any risk on quality, efficacy, or safety of your drug product after the modification, you have to follow the stepwise approach for therapeutic equivalence as described in the section 5 of the guideline. And this is very important also for the post-authority changes. You have to follow exactly this therapeutic equivalence pathway. So the key messages here are the demonstration of therapeutic equivalence is a stepwise process. IVRT is always an essential part of the process, and the results of an IVRT can be overridden by the next level test. And important, the stepwise approach is also binding for post-approval changes. Method development and validation. In Annex 1 of the guideline, you will find the information about IVRT method development, validation, and the expectations of the authorities how to present these data. So, in general, you have to provide a summary of the method development and the data of a proper method validation to demonstrate that the IVRT method and the analytical method is fit for purpose. Please keep in mind that regarding the guideline, for method validation and for equivalence testing, you always need 12 replicates per batch. Yeah. Only for routine testing, for example, during the stability, you need only 6 replicates. And this is in difference to the FDA, where you always need only 6 cells. The reported IVRT data should always contain, according to the EMA guideline, the drug release rate, as well as the cumulative amount and, if present, the lag time. When starting the method development, from my perspective, IVRT method development should always start with a receptor medium selection based on the API solubility. According to the guideline, sink condition is required, and that means that the concentration of the API at the end of the experiment is 3 to 10 times below the maximum solubility of the API in that receptor medium. That ensures that you don't have any rate limiting effects due to a high concentration in the receptor medium. You can add organic solvents to increase the solubility of the API. For IVRT, it is not necessary to have a physiological solvent like an IVPT experiment when you typically use a phosphate buffered saline. This is not necessary for IVRT. Hydroalcoholic solutions are used very often. And from my point of view, and my recommendation is, when using organic solvents, try to use organic solvents which are typically used also in the analytics to avoid any interference later on during analytical assessment. Besides this, try to use always a buffer instead of pure water to keep the pH constant during the experiment. In the table below, there's just an example for a screening. There are two different organic solvents in citrate buffer and the effect on the solubility of a specific API. Choose the receptor medium or the organic concentration should only as high as necessary to achieve the solubility to avoid any negative effects on your drug product by back diffusion. In addition, you have to prove that your API is sufficiently stable in the selected medium, at least for the duration of the IVRT experiment and the following analysis. And please ensure that the receptor medium is compatible with the membrane. Having a receptor medium in place, the next step will be the membrane selection. For IVRT, only synthetic membranes are used, and there are a lot of hydrophilic and hydrophobic membranes commercially available from many different manufacturers. Typically, a pore size of 0.45 microns are used, but this depends on your drug product, and other pore sizes, a little bit smaller, a little bit higher, wider, are possible. For example, if you have a very thin lotion, 0.2 microns could be more suitable instead of 0.45 to keep the drug product in the donor compartment well separated from the receptor medium. Start the membrane selection by testing non-specific binding of your API to the membrane, and use only membranes with no or almost no absorption effects. Then conduct an IVRT experiment with the potential membranes and choose the membrane with a good linearity and the highest cumulative amount, because this means that you have the lowest rate limiting effect. For IVRT, a pseudo-infinite dosing is requested by the authorities to avoid a significant depletion of the API from the donor compartment, which could have an effect on the release rate. So test the type of application, and I would recommend to use a positive displacement pipette to ensure a good application with a low variability. Cover the cells to avoid evaporation and any other effects which can cause a transformation of the applied drug product. So in contrast to IVPT and IVRT, you have always covered front cells. The sampling is expected at least hourly, with at least 6 data points within the linear range. A higher sampling rate and a longer sampling time is recommended for method development. You can shorten it later on in the validation. But for method development, I recommended a longer period to get a better understanding of your drug product. And of course, your IVRT is only valid if you are able to demonstrate a linear drug release. The experimental temperatures, or the experimental temperature is almost fixed. Use 32 degrees for topically applied and 37 degrees for products applied inside the body, like a rectal or a vaginal product. The mixing rate depends on the equipment. There's no fixed value, but the mixing rate should be sufficiently high to ensure a fast distribution of the API in the receptor medium to avoid any higher concentration below your membrane, which could also affect the release rate. Having a well-developed method in place, you can start the validation of the IVRT method. And the very first point of validation is the validation of your analytical method. So analytical method validation should be finished before IVRT method validation can start. The aim of IVRT method validation is to demonstrate that the developed method is robust, that's clear, and discriminative against modifications in the critical quality attributes of your drug product. There is no explanation in the guideline how to demonstrate linearity, intermediate precision, intra and inter-run precision. It is just mentioned that we have to show it. From my point of view, I would recommend this procedure described here. This is based mainly also on the instructions given by the FDA. So run 3 independent IVRT runs with 12 replicates and use 1 single batch of your formulation. With this data, you can calculate the linearity and intra and inter-run precision. And when you use 2 different technicians for these 3 IVRT runs, you can also use these data to calculate the intermediate precision. And an acceptance criteria is only given for the intermediate precision in the guideline, and this should be, or this should have an percent RSD of below 10% between 2 different technicians. In addition, the demonstration of specificity is expected in the guideline. And this means that you have to demonstrate that the release rate is a linear function of the API in the product. At least 3 different concentrations should be used, and the R-square greater than 0.9 of the linear curve is requested by the EMA. Although not explicitly mentioned, try to use 50, 100, and 150% of the nominal concentration. I know 150% can sometimes be challenging, but this is recommended from my point of view. Besides specificity, you have to demonstrate the discriminative power of your method against changes in critical quality attributes. So the equivalence test must be negative when comparing the altered formulation with a reference. This is not trivial, and the effect of formulation changes on the drug release rate should be evaluated before the validation process starts. Finally, the robustness should be evaluated to ensure that small changes in the setting does not affect your results. Sounds simple, and it is, but the robustness test dramatically increase the cost of your IVRT validation, because you have at least 8 extra runs with 12 replicates each to conduct, just to demonstrate robustness. So, last but not least, data evaluation. The two one-sided t-test is used for equivalence testing, and the 90% confidence interval for the geometric mean ratio of test and reference should be within 90 to 111%. On this and the next slides, I show you the required formula to calculate the equivalence test. In general, the equivalence test based on the differences between two groups and cannot handle ratios as requested or as required in the guideline. But that's no problem. A log transformation of the data can be used. So the ratio of the test and the reference product on the linear scale is mathematically equivalent to the differences on the log scale. So now you can calculate your statistics with your log transformed data. The calculation of the 90% confidence interval can be done with this formula. So t-value depends on the number of your samples and the requested confidence interval, in this case the 90%, and can read from a table or calculate it in R or Excel. And the pooled standard deviation, here SP, calculated by this formula, takes the variability of both groups into account. And with these values, you are able to calculate the 90% confidence interval, the lower and the upper bound. And then the last step is the back transformation of the lower and the upper bound from the log scale to the linear scale, and to check if these bounds are within the 90 and 111%. If so, you can be happy because you passed the equivalence test according to the EMA guidance. To be honest, 90 to 111% is a very tight limit and much tighter than the FDA acceptance criteria, which allows 75 to 133%. This was discussed since the draft version of the guideline was published in 2018, with the result that the acceptance criteria can now be widened up to 80 to 125%, depending on the variability of the reference product. An example of the limits in accordance with the observed standard deviation is given in the guideline and not explicitly mentioned here in the presentation. Unfortunately, there is no clear recommendation in the guideline on how to determine the variability of the reference product, and the extended limits here from 80 to 125% are still below the standard acceptance criteria of FDA. So at the end of my presentation, I can conclude that EMA and FDA missed the opportunity to harmonize the guidance and the acceptance limits. In addition, EMA guideline is more or less a top level and not a hands-on guidance, so a deep expertise is needed for proper IVRT method development and validation. Although the revision of the guidance took 6 years, there are still questions left on how to interpret the IVRT data in the real world. Just an example, IVRT is expected to be part of the stability program, but how should a drift in the release rate over shelf life interpreted? What is a relevant drift? Or therapeutic equivalence should be demonstrated with at least 3 reference and 3 test batches. How do you compare the batches? Do you compare batch by batch to get 9 results? And does each result has to fulfill the requirements? That's not clear. So the guideline is new and we all have to handle it. Some questions will only arise in practice and have not yet been considered. So the best way is to work closely with the authorities and use scientific advice to clarify open questions in time. So the take-home message or my presentation in just 6 bullet points. IVRT is of increasing importance in topical drug development. Take care of it. Drug release is a critical quality attribute which should be specified and controlled, and the specification limits based on clinical trial batches. So you have to generate the data before commercial manufacturing. IVRT is part of your stability program and relevant for scale-up and post-approval changes. And finally, IVRT is part of the stepwise approach to circumvent clinical trials to demonstrate therapeutic equivalence. So, thank you very much for your attention, and I guess we will have some time left for the Q&A session. Thanks a lot.
Thank you, Sasha, for this excellent presentation. So we will now move on to the Q&A session. I would like to remind everyone that you can submit your questions using the question function on your screen. So Sasha, let's see, we have a number of questions already coming in here from the audience. So the first question is, you mentioned that a formulation with 150% of the nominal API concentration should be used for demonstration of specificity. What if this cannot be dissolved?
Yeah, good question. I see the point. For a formulation with a high saturation of maybe approx 80%, or it's a nominal API concentration, 50% more API cannot be dissolved, and the API will partly suspended in the formulation. In this case, it is really difficult to predict how this affects the release rate. The release rate can be under proportional low, as only dissolved API or can contribute to the release rate. On the other hand, you have a 100% saturation, and this can lead to an over-proportional increase of the release rate due to the high thermodynamic driving force. Difficult words, sorry. So in general, although required in the guideline, this demonstration of specificity, a linear relationship between concentration and release rate cannot always be assumed and depends on your formulation.
Okay,
so I hope
thank you, Sasha. Next question. To demonstrate discriminative power of the methods, critical quality attributes of the formulation should be changed in order to change the release rate. Can you tell us which attributes should be changed?
Different formulations can have different critical quality attributes. So in general, it is important to have a good understanding of your formulation design. At Vardes, we have formulation experts who can help with this decision. one option can be the change of the most important solvent for your API. This will have a significant effect on the saturation level, which usually has a direct effect on the release rate. So this can be a critical quality attribute which can be changed.
Okay, thank you. Next question. Do you use the Phoenix RDS system, which is the Teledyne Hansen system, the robotic diffusion station? And do you think this has advantages over the traditional front cell?
Yeah, a clear yes. The robotic system allows us continuously sampling over 24 or more hours, which can be necessary when assessing the slow release from an ointment. And this would not be possible using a technicians because usually they don't work during nighttime. This is really an advantage. And besides this, the dry heat system to control the temperature here reduces the variability between the different cells in comparison to the use of a water bath, which typically supplies the cells sequentially with pre-warmed water, and this caused a temperature shift from the first to the last cell and probably increase the variability. So yes, yeah, this is a really advantage to have the system here at Rades.
That's good. Happy customer.
Yeah.
Next question. So you said the IVRT is a performance test, but in reality, IVPT is actually considered a performance test, and IVRT only use filter membranes which have nothing to do with the skin barrier. Why do you consider IVRT the performance test then?
Because the EMA guideline uses the term performance test also for IVRT. But you're right, it is not considered to be predictive for efficacy or skin penetration. Yeah, IVRT only is for technical performance and cannot assess any biological effects. So it's maybe better understanding technical performance of your formulation.
Okay, great. There are a few more questions. I think we have a few more minutes. So next question. You mentioned that the acceptance criteria of an IVRT depends on the variability of the reference product. Does it make sense to generate as much variability as possible when determining the IVRT data for the reference product in order to expand the acceptance limits later on?
Good question.
Complex question.
Good question, but my understanding is that the reference product variability corresponds to the variability between different reference batches. So when you compare batch 1, batch 2, and batch 3, and not the variability observed within the results of a single IVRT run with 12 replicate cells. So therefore, from my point of view, it makes no sense to artificially increase the variability of the reference product.
Okay. Next question. So what are the main differences when comparing the EMA guideline with the FDA, the USP guideline for IVRT? And then you do mention in the presentation about the opportunity the missed opportunity of harmonizing those guidelines. So why, in your opinion, they didn't take this opportunity to harmonize them?
In general, a good question, and we could do an extra presentation about this topic. But the main differences here in IVRT are the acceptance limits, the kind of statistical evaluation, and the amount of replicates needed to demonstrate equivalent drug release between the two formulations. I'm not sure why we see such a huge discrepancy between both guidances, and yeah, I cannot explain why we have this discrepancy here.
Okay, well, if anyone here in the call have any answers, drop us an email and want to find out.
I would also prefer a harmonized version between both agencies.
Everyone, I guess, in the industry, it would be much easier for sure. Especially for multinational companies when they have maybe US-based sites, European sites, and so on. That would make everyone's life easier, I guess. Okay, so next question. If there are differences in Q1 to Q3, so the qualitative, quantitative, and microstructure, can we still have a pharmaceutical equivalent product?
No, because Q1 and Q2 compares if you use the same excipients at a similar concentration. And if you can't demonstrate at least one of this, you don't have a comparable product, and this is per definition not pharmaceutical equivalent. So no, if you have Q1, Q2, and Q3 different, you can't demonstrate pharmaceutical equivalence.
Okay. I think, we're up here with the time, so that will bring us to the end of today's webinar. Sasha, thank you very, very much once again for your presentation, for sharing your expertise here with us today. Of course, to all of the participants, thank you for joining and for engaging, for asking so many questions, great questions, by the way. So as mentioned earlier, a recording of this session will soon be available on the Teledyne Labs YouTube channel. Feel free to revisit this content or to share with any colleagues or people in the field who may find it valuable. For more information about this webinar or to explore additional resources or products from Teledyne Hansen, please visit teledynelabs.com or connect with us on social media. So we truly appreciate your time, your interest, and we look forward to welcoming you to our next webinar. Thank you.